tutorial, and we're going to post this. We couldn't get it live, but we're going to post it later. So right now we have Tom Portuguese, and I mentioned about Morphozoic during the Flash talk, and I didn't really do it justice. So he's going to show us a little bit about Morphozoic. So Tom, take it away.
Okay, hi, I'm Tom Portuguese, and so this is my the Morphozoic talk tutorial. So Morphozoic was written as a chapter in a book. You can see it on the screen there, to appear in February 2017. And so the title is Cellular Automata with Nested Neighborhoods as a Metamorphic Representation of Morphogenesis. Quite a mouthful, but we shall determine what that is. And there's the authors on the bottom. So Bradley and I are on the author list, and a couple other people, including Dick Gordon and Gabriel Pasquale and Steve McGrew. And there's my email also included. So Morphozoic is an abstraction of morphogenesis. So what is morphogenesis? Well, and also, how is this related to DevaWorm? Well, it's related to DevaWorm because that's basically DevaWorm's rationale for being. I won't try and say that in French. Is how C. elegans develops from a single egg to an organism, fully fledged organism. So morphogenesis, this is basically scraped out of Wikipedia. It's a two-part word. It means, the Greek word morph means shape, and genesis means creation. So it's a biological process that causes an organism to develop its shape. And so what this means is how the cells in the organism arrange themselves and change from an embryo, from a single cell egg, fertilized egg, to an organism. Okay, so nature has found a lot of ways of doing this. It uses chemicals, it uses hormones, which are, of course, chemicals. It can use mechanical stresses. It can use lots of ways of signaling cells to do something, to change into other patterns of cells. And so what we're interested in is kind of abstracting this, finding a computational model that incorporates some of the richness of how this works. And we're going to use a thing called the cellular automata, which will be explained a little bit further on. And we're going to be using things called nested neighborhoods within a cellular automaton, to be explained further. And just like in morphogenesis, we have some influences that come from far away, and they're more diffuse, more global, you might think. And some influences are local, like a mechanical stress, like bumping into something. You might think of that as more of a local, immediate influence. So we want to have we want to be able to incorporate both these things, local and global. And also we want a model that's actually not going to go off the rails computationally. So this one does that by basically aggregating cell values. And what it does is it allows the thing to actually become more complex in a linearly growing fashion, not geometrically. Okay. Cellular automata. So this is a very fundamental computer science computing structure, and it's very simple. It's a two-dimensional grid. There's variations that could be 3D, but the most common one is a 2D grid of cells. So each cell has a state, which is just a number. It could represent anything. We're also going to call these states types later on as a cell type. So each cell can not only knows what its own state is, but it knows what the states of its neighbors are. Well, what is it? What are neighbors? Well, a very typical neighborhood, as it's called, defining what a cell's neighbors are, is 3 by 3, a set of cells surrounding a cell in a grid, and that's called a Moore neighborhood. And a cell can change states, and it will change states depending on what its own state is, what the states of its neighbors are, and the set of rules. Okay, probably the most common instance of a cellular automata is Conway's Game of Life. So you can see there's the grid. It has black and white cells on it. The black ones are called alive cells, living cells. The white ones are dead cells. And Conway's Game of Life has 4 rules. So if you're alive, meaning black, and you have fewer than 2 live neighborhoods, you die because you're lonely. It doesn't matter which ones of your neighbors they are, but you'll die if you don't have more than 2, or fewer than 2. If you do have fewer than 2, sorry. Rule number 2: if you're a live cell and you have 2 or 3 live neighbors, you just get to stay alive. You're kind of happy with neighbors. It's the acceptable number of neighbors. Any live cell with more than 3 live neighborhoods is overcrowded and you die. So the first 3 rules have to do with live cells either staying alive or dying. So how do we get more live cells? Well, we have to have a reproductive rule. And here it is, the last rule. A dead cell can come to life if it has exactly 3 live neighbors. Now, with these simple rules, and I encourage you to go look this up because it's really super fascinating, you can come up with amazing patterns. You can actually build a full-fledged Turing machine with this stuff. You can compute anything. What you see on the screen here is just a bit of some random stuff. You can have things that reproduce different patterns, things that fly across the grid. I can't begin to even tell you. Go look it up. It's worthwhile. So we're going to have also not the game of life, but a different sort of rule set in a cellular automaton. And so again, our cells will possess states, also called types. A cell will emit, sense, and react to signals, so there's some kind of exchange of information between cells. And the signals actually, the purpose of a signal is to actually transfer the state of neighborhood cells. So basically that's how cells find out what other cells are, what state they're in. And a field is a confluence of signals sensed by each cell. So the idea is that a morphogenetic field, as it's called, is a superpositioning of neighborhood signals about their states. We want to be able to have a compact, noise-tolerant, generalized, and evolvable. Compact has to do with the linear growth and complexity. Noise-tolerant has to do with we don't want little glitches here and there, fuzzy bits of noise to throw the whole thing off. The Turing example we're going to look at in detail, actually, that really is important. And we want to be able to have some kind of generalization. Basically, we want to be able to start in some state that's similar to another state and actually do something sensible instead of just being completely inert. Nature, of course, uses evolution to change things. So we want to be able to actually evolve, model some kind of evolution. Okay, so another term, morphogen. So morphozoic has this thing called a morphogen, and this is an actual term borrowed from biological morphogenesis. It's basically the input to the process. So basically, in a cellular automaton, it's the neighborhood states. And we're going to do this thing called nested neighborhoods. Well, nested neighborhoods means like Russian dolls. Little neighborhoods are part of bigger neighborhoods, which are part of bigger neighborhoods, which are part of bigger neighborhoods. You can grow indefinitely, linearly, as I said, which is nice. So we have to start with some neighborhood, of course, and in this case, the lowest, smallest neighborhood is a single cell. We're going to call it an elementary neighborhood. Non-elementary neighborhoods are bunches of cells. They're actually n by n sets of cells. And each one of the regions in this n by n is called a sector. Now you're wondering, why don't I just call it a cell? Well, because in larger neighborhoods, these sectors could be composed of multiple cells. You'll see a picture. So we're going to call them sectors because they're aggregations of cells for larger neighborhoods. And each sector, what we're going to do in each sector, it could contain multiple cells, a little subgrid of multiple cells. We don't want to actually incorporate all that information into the value of the sector because that's going to blow up and get too complicated as neighborhoods grow. So what we're going to do is just go through the sector, this little n by n grid, count up all the cell type densities, count up all the cell types, and make a histogram. That's the value of the sector. Okay, so we're basically we're aggregating the information in the sector cells. Here's a picture. Hopefully this makes more sense. So on the left, we have a grid. We have a bunch of cells. Some of them are white, some of them are black, some of them are gray. I think there's 4 different flavors there. So that one cell that the orange line points to is the most elementary neighborhood of that cell. So that's neighborhood sub-0. Now if we add a couple of layers of neighborhoods, basically we're nesting this thing. We can put that cell in its neighborhood, next higher neighborhood, which is neighborhood sub-1. That's 3 by 3 in this case. Now morphozoic's more flexible. It doesn't have to be 3 by 3, but it's 3 by 3 in this case. So that's neighborhood sub-1 is a 3 by 3. And each one of the sectors in that 9 sector neighborhood happens to be a single cell because it's the smallest non-elementary neighborhood. And right below here, you see we have a little histogram of we count up all the different types of cells, and we make it a little histogram. Let's move over here on the right-hand side. So now we have a 3 by 3. Let's make a 3 by 3 of 3 by 3s. So this is neighborhood sub 2, which nests neighborhood sub 1, which nests neighborhood sub 0. Here we can also do the same histogram trick. So we go ahead so here's 9 sectors in neighborhood sub 2. For each one of these sectors, we're going to go ahead and count the different cell types, and you can see a little histogram here. And that trick, it's not the only trick for aggregating these things, allows us to kind of collapse the amount of information in here to some kind of manageable amount. So now we have we have got this thing called a morphogen, which is the input to the process. So a morphogen describes what we've got. So a single cell can sit there and go, oh, I've got this morphogen sitting here. It's a bunch of nested neighborhoods surrounding me, and it tells me in a nested sort of local to global way what my state is, what my super state is, so to speak. But we need to actually change things, cells, states or types. And to do that, we need this metamorph. So metamorph is a simple thing. You put a morphogen in as the input, and then you have an action rule. And all the action rule is change to a cell type. So if a morphogen, if a cell matches a morphogen in a metamorph, it will change to the cell type that the metamorph tells it to change to. So it's a state action rule. Okay, now we found it very beneficial and we get some interesting and useful effects by using an artificial neural network to train metamorphs. So if we're going to look at a bunch, so we're going to have run different configurations of cellular automata. They're going to generate a lot of metamorphs, which contain morphogens, and we want to learn which morphogens should cause certain transitions to cell types. And a useful mechanism for compacting this instead of having a huge database is train a neural network to do this. Once it's trained, it takes a while to train it, but once it's trained, it's super fast. So basically, any cell can go, oh, here's my morphogen, plug it in to the neural network, and out comes its cell transition. So we train this by taking the morphogen, which is this nested n by n thing, and flattening all that stuff out and stuffing it into the inputs on the left-hand side here of the neural network. And then through the magic of training of a neural network, which I'm not going to discuss here, we can have the neural network figure out which cell transition should occur. So now once we do that, we've got this really nice little box here that we can quickly, and the beauty of this thing is it's noise tolerant too. And that's going to be super important later on for the example we're going to show. Also generalizes, which is also super important. Okay, so in the chapter we decided to do this and that to kind of demonstrate different features. And of course we should baseline it, so to speak, with Conway's Game of Life, which we did do. So what does that mean? Well, we played some games of life. We started out with certain input starting configurations, ran it, Morphozoic watched it, generated metamorphs containing morphogens, and we trained it to be able to play the game of life. So it can learn, essentially, specific scenarios of game of life by watching game of life scenarios. We also taught it how to do cell regeneration. So in the chapter, you'll see like images of this famous image of Lena. I think it's called Lena, where we started out with certain pretty sparse patterns of Lena. That's a woman's face, and it can regenerate the image by simply watching how it's done. We also did evolution, so a simple gas relation sequence, so it can actually figure out what the rules are by kind of trial and error process with the genome and so on and so forth. That's kind of out of the scope of this particular talk, but it's in the chapter. We also can do, as a demonstration of generalization, neurons in the brain during development try and find other neurons to connect to that they're supposed to connect to. So they use signals to do this. So what we can do is try and model this with Morphozoic, and it can find basically neurons can seek out other neurons that are nearby from signals in a very generalized manner. So they don't have to be taught specifically, this neuron's here, that neuron's there, send an axon over to that neuron. It can use these signaling cues to do that. It can learn to do that. The last thing is the actual demo we're going to do, which is a simulation of Turing's reaction diffusion morphogenesis. Here it is. So, Turing came up with a scheme for a mathematical scheme using differential equations to take what looks like kind of randomness and produce patterns that are kind of recognizable as some kind of biologically plausible patterns of something. So here is a cellular automaton version of this. So on the left, this is a random pattern. On the right, it looks like something like a cheetah code. This is actually known as the cheetah code pattern. So his process will take a random left hand, change it into the orderly right hand. How does it do this? Well, here's the paper, 1952, and sort of historically, for historical significance, this was his last paper before his tragic demise. And I also included a little bit of background about it from Wikipedia. So you can, his process will create stripes, spots, spirals, things that you seem to, that people intuitively see in biological organisms. Not just the cheetah pattern. And it's called reaction diffusion, is the general term for this. Here's the equations for a two chemicals system. This is the one we're actually going to be using. This is the one that creates the cheetah pattern. So if you have a chemical U, chemical V, the chemical U and the chemical V will change in each cell depending upon the levels of concentration of these two chemicals in the cell. And there's a couple parameters here that if you set these differently, if you set these one way, you'll get a cheetah pattern. If you set these differently, you might get a spiral. But the same equation, different parameters. So the three components are called production, degradation, and diffusion components of the equations. Now, what we're going to do is expose Morphozoic to this and see if it can learn what's going on. So Morphozoic is going to take a random pattern, not the same random pattern, a different random pattern. This is where this neural network comes in really handy, because it can actually sort of settle into certain patterns that it learns. Okay, and produces a cheetah pattern, and you're going to be able to see this in the demo. Okay, let's get into the nuts and bolts here. So, to do Morphozoic is written in Java. So you need Java, and you can get it. You need not just Java, but you need the JDK. So the JDK has the compiler in it. So that's what you're interested in. So Java's owned by Oracle. Go to that website and download it and install it. You know, obviously it can run on any platform, Windows, any kind of Linux, you know, Macs. Okay, you want to go now to Morphozoic, which is, there's the link, it's in GitHub, and I would just grab the zip file. You don't have to clone it. If you want, you can clone it if you like. Okay. And now you can follow a couple of simple instructions that I'm going to run through for you. So it includes an Eclipse project if you like Eclipse. I like Eclipse. A lot of people like Eclipse. Eclipse is really good for Java development. You don't have to use Java Eclipse, but you can. So we're going to do it manually though. So we're going to take the zip file, unzip it. We're going to go to this work directory, and we're going to run build.bat, because I'm on Windows here. But if you're on Linux, you can run the build.sh. And I'm also going to run a couple of sh commands. Even though I'm on Windows, I have a bash Cygwin shell. The first one I'm going to run is actually the code for the actual original Turing morphogenesis. And Morphozoic's going to be watching while it does that and learning and putting it in a save file. And after that, I'm going to run Morphozoic, train a neural network, and see if it can actually reproduce what it saw the Turing morphogenesis do. So, okay, let me pop out of here then. And go to, I've got this thing already, oh, okay, so where is it? It's right here. That's the link. It's on GitHub. Just go ahead and grab a zip file here, or clone it, whatever. And so I've already done that. So there it is. I already unzipped it. And I'm going to go using my bash shell. There's actually two sort of sub-projects here, but you want the one, not the polar charge cells. That was kind of more an original 3D version of this, but it's there. That's C++ actually. So now you go and there's a work directory. It has some scripts. So the first thing you do is build it. I'm going to build it with bat because I'm on Windows. Okay, built it. Now you see there's a bunch of scripts in here to run various applications. You can run the other ones later if you like, but we're going to do the Turing ones. So I'm going to run the Turing gen.sh. And it's thinking, and it's going to randomize a bunch of cells. Okay. So now I think I can just step it a few times. Step. Crunch, crunch, crunch. Takes a while. Oh, there's something. Let's step it again. So the parameters for this are set to do the cheetah. If you fiddle with the code, you can get some other patterns. We won't do the cheetah, overkill the cheetah. I mean, it's already looking cheetah. So we'll do one more step. So Morphozoic is watching this thing. It's going, ooh, now that's a decent pattern. Okay. So let's just get out of here. And what it did was it stored a database called g.dat. You can see it in the g.dat is the file that it stored it. So now we're going to actually see if Morphozoic learned how to create the cheetah pattern. And that's this script, turingexec.sh. And this is going to take a little while to come up, I think, because at this point I think it's training the neural network. Won't take too long though. Here we go. When it pops up, it'll be ready. It's almost coming up. Okay. You step it. So here's a different random pattern. And now the neural network's going to see, oh, like this cell here, what is its neighborhoods? What do they look like? I think it has like the same, I think it has 3 levels of nesting.
Oh, wow.
I think. Could be wrong, but I'd have to look. Oh, maybe it's in here, actually. Oh, it has, 3 neighborhoods. That's what it's using. And I think it's 3 by 3, but I'm not sure. So let's step it again and see if it's making any progress towards producing a cheetah pattern. And it's crunching. It's actually, it doesn't look very cheetah-ish. Let's see if it can get better. Oh, it's slightly better, maybe. Getting better. Maybe 1 or 2 more. Well, maybe 1 more. It's taking a little while here. Okay. All right, 1 more. If I let it run further, it would get better. But in the interest of time, we'll. Okay, we're gonna call it a day here and say that's a cheetah pattern. it'll get better, but we're not gonna sit here and watch it. Okay. So that's how you can actually run the thing, and obviously the code's all there. Okay, so I've got a couple extra minutes. I wanted to plug another project very briefly. If we take this concept of global, local, global meaning distant in space and time, local meaning like recent and very less distant, can we actually use that to compute things, not just morphogenesis? So basically, can a brain kind of do this? So here's the idea. Is a brain, so we want to know how intelligence works, we look at brains. But a brain is a solution to a problem, not a problem. Also, it's made out of cells because nature makes stuff, everything that's its building block, so it makes stuff out of cells. So can we agree, though, and I think we can, if we're not philosophers, that out there in the world are dimensions of space and time, and events happening in space and time. And that's what this thing is built upon, that assumption. So this morphognosis is the project name. It's called, once again, morpho for shape and gnosis for knowledge. Basically, it's the shape of knowledge. So it's a pyramid, and it's going to look like a pyramid of space-time chunks, which we're going to see in the next picture. So at the apex are the most recent and nearby events in space. Farther away on the pyramid, down the pyramid, are things that are less recent, I mean more distant in the past, and more dispersed in space. Here's the picture. So basically, it's the same sort of nested neighborhoods thing that we saw in Morphozoic. Far away, so things in the past, lots of possible spatial distances. Here's a little snapshot of the app running. There's a critter in here called a MOX, not related to uranium ore or reactor ore fuel. It has to do with, it's historical. I'll shut up about that. Okay, so this little blue thing is the MOX. It's a creature. It has an orientation you can see because it's pointed in a certain direction, so it can be pointed up, down, left, right. The green circle is food. The other non-white things are obstacles, so it can sense the types of these obstacles. The white squares are just empty. It can move around on them. And you can see a little dashboard here. If you click it, you get the dashboard popped up. So what we can do is do the nested neighborhood trick. And here we've got the layer 2, which is 2 beyond the most local. And we have a 3x3 set of sectors. So it's the neighborhoods. The neighborhood is a 3x3. And we have this little histogram deal. So basically, this is a chunk of space-time with a histogram of the information that was in that chunk of space-time. And we're going to use that to learn what to do to find the food. And here is, it's not going to actually use this, but this is the source of those histograms. So again, it's a 3x3 set of cells. So this represents, this is a little picture of what it saw in the past, and it dispersed distances. And so that is actually an ongoing project, which is working on, and that is my presentation. I might want to give us, since I've got a couple of minutes here, give a sneak peek. I'm also on the OpenWorm Education Committee, so I'm working on getting OpenWorm a presence in the mobile arena. And what we're doing is building an educational app to allow students or interested parties to train a C. elegans to find food. And this is based on a couple of technical papers, one by Beer and Izquierdo and Beer, and the other one by Netta Cohen and a couple of other authors. So I'm using Marmalade to build this. I'm going to show you how C. elegans. So this is a simulator of a phone. So this is going to run on Android. It's going to run on iPhone. It's going to run on the Windows Store. So what you see here is I'm squeezing the screen. You can't see me do this, but okay. So over here on the sort of right-hand side is the worm. Here's some food emitting a salt. C. elegans has the ability to sense salt in its world. And so that's a little bit of, that's a guy he wants to eat because it's emitting some salty signal. We can actually also look inside the worm here and see its muscles. Just its muscles, doesn't have any other innards. And so we want the worm to actually try to find and eat this thing. It can't do it right now. But we're going to be able to, these are neurons, these are the steering neurons within the worm, the actual worm, and the names of these neurons that control how it senses salt in its environment and how it turns itself left, right, how it can steer itself undulating in its environment to get to that food source defined by the salt. So the trick is you're going to be able to take these little bubbles here, which are the, I know this is all very quick, but you're going to be able to weight these connections between the neurons such that they'll signal each other to become more active or not. So for example, I can do this one and I will be able to change the weighting on that synapse. The synapse is a connection between two neurons. And I'm going to be able to, let's see what happens when I run this thing. Obviously, it won't be able to find the food at this point because that's your job. you can see it trying to, it'll just go past the food. You can see it actually, the neurons firing here, and it's steering motor neurons. So it kind of headed off in the wrong direction, didn't get to the food, but that's obviously the trick. And you can see its muscles once again stretching and compressing. So that's going to be a mobile app for students and interested parties to have a little bit of fun figuring out how to actually modify the neural network of the C. elegans in a very sort of biologically plausible way. It's all very scientific here with these reputable neuroscientists doing this, and that will be forthcoming before the end of the year, I think. So that concludes my talk. Thank you very much, and back to you, Bradley.
All right, thank you, Tom. How long do you have before you have to leave us?
I got a few minutes.
Okay, maybe we could run through some of the questions I had,
Sure.
or observations.
Sure, absolutely.
So the first part...
Maybe I'll get better answers.
Well, it's okay. So the first part, if you could go back to the slideshow.
Okay.
Bring it up on the screen.
Here we go.
Yeah. So let's go back to the Game of Life slide. All right. So the first observation is that if you look at the Game of Life and you look at what's going on, some people, maybe like biological cyberneticists or someone like that, would argue that there is a complex autoregulatory system at work here, that the cells are regulating each other in communities. And furthermore, some people might argue that that's there's something called biological decision-making going on. That's, one view. But it's interesting how the cells are actually, or the system, whatever that is actually learning rules of interaction and creating order from them. So maybe can you speak a little bit to that observation?
Yeah, well, for the Game of Life, that's kind of why this is fascinating. These four rules that don't seem very complex, are the basis of some really complex, really intuitively interesting patterns here. So, so you can get things like little flying things and reproducing things and so on with this. So in nature, this is always the fascinating questions, like how did nature evolve certain rules that cells abide by to sort of regulate themselves in a sensible way such that they survive and reproduce, Yeah. So that's actually, that's actually not a topic within the scope of Morphozoic, but it's just in general one of the, what are the basis of morphogenesis?
Right.
How do these rules come to be? These chemical reactions, such that cells can auto-regulate, as you say, and produce organs and tissues that actually do something sensible, and produce a worm that can live quite happily in its environment. That's part of the amazing stuff that, as a computer scientist, this is just incredible food for thought.
Yeah.
So I know it's not a very technical answer, but it's a rah-rah answer.
Oh, that's fine, yeah.
Maybe you can comment on that, Bradley, because you're more of a biologist.
Well, we can talk about that later. But
yeah.
Next, go down to the metamorph, or where you have the. No, down a little bit more.
Okay.
Yeah, there we go. So yeah. So I guess I'd like to point out that this is a nonlinear. You said it was a nonlinear mapping between inputs and some sort of cell type.
That's the beauty of artificial neural networks, is they can do nonlinear functions or linear functions. They are quite happy to do both of those.
Yeah.
So if it turns out that there's some kind of nonlinear I mean, some inputs are more important than other inputs, and maybe they don't. Like there's maybe some kind of Gaussian, bell-shaped function involved with the inputs and the outputs. It'll learn that.
Yeah.
So it's quite happy doing that.
Also important to point out that in C. elegans we're dealing with like a deterministic form of differentiation, but in many organisms you have many signals that have to be sorted through, and then there's some decision made that's within a context. So it's not deterministic, but this sort of model actually allows us to really kind of get a grip on that without really being too hard to define or combinatorially explosive. So
yeah, this is quite compact. Also, like speaking to the non-deterministic thing, I mean, a neural network will do a best match, and so in the outputs here, you can see on the right-hand side, it will produce essentially a probability or a strength or a confidence factor for each one of these cell states that it should transition to.
Okay.
You can then make a probabilistic decision as to what the cell should change to.
Okay. Yeah.
So it's context because you have the nested neighborhoods, and it's also probabilistic, so it's not totally deterministic.
Yeah. So a final question I had, if you go up to where we talk about the nested neighborhoods and the cell densities. Yeah, right here. Yeah. So
okay.
I wanted to point out that this is a sort of a strictly hierarchical system where you have nesting of complete sets within complete sets.
Mm-hmm.
But in some cases, you might not have that in a biological system where you might have a strict hierarchy where you have things that are hierarchical, but they're not nested necessarily.
Yeah, they aren't strictly, Russian dolls, right?
Right.
Of some sort. Yeah, certainly. Yeah, that's the beauty and the limitation of this is that it's something it's computationally kind of you can analyze it. It's tractable, So when you get into nature, it's like all holds are no holds barred, right?
Yeah.
It can be really fast. we even as a non-biologist, I've been reading, how many things in C. elegans are that seem to be that way.
Yeah.
It's just great. In this tiny 1,000 cell organism, you've got an amazing number of interactions going on that aren't exactly nice and clean,
But this is again a probabilistic model, so we can.
Yeah, this can be. This is definitely can be a probabilistic. You can be as probabilistic as you like. It's a tunable parameter, Morphozoic.
Okay.
So.
Yeah. Well, good job. Thank you very much for giving us this tutorial.
Yeah. You're welcome.
Okay. See you later.
So thank you again.
Okay. Anyone can reach you on our website through our website.
Yep.
So okay.
Yep. And I've got my email on the title page.
So. Okay. All right.
And if you're interested, I can send you the slides.
All right.
Okay.
Okay.
Next time.
Thank you.
Bye-bye.
Bye.